Good morning to all. Welcome to all of you in this video lecture. In last video lecture, we discussed on fertilization part first. In this way, in this video, we will discuss on fertilization part second. MSc फाइनल बॉटनी पेपर फिफ्थ के इस वीडियो में अपन फर्टिलाइजेशन पार्ट सेकंड के अंदर कुछ पॉइंट्स पर डिस्कस करेंगे। वो इस प्रकार है। फर्स्ट ऑफ ऑल, एंट्री ऑफ पोलन ट्यूब इनटू द ओव्यूल। पोलन ट्यूब की एंट्री ओव्यूल में किस प्रकार होती है? आफ्टर अराइविंग इन द ओवरी, the pollen tube finds its way into the ovule. Depending on the place of pollen tube entry into ovule, fertilization is of three types. First is porogamy, second chalezogamy, and third mesogamy. Porogamy most common type, in which pollen tube enters through micropyle. Chalezogamy, pollen tube enters the ovule at chalezal end. Example, Casuarina. Mesogamy, the pollen tube entry into the ovule is through funiculus or through integuments like cucurbita. Then entry of pollen tube into ovule and its subsequent growth toward the embryo sac is regulated by a chemotropic substance secreted by the filiform apparatus into the micropyle. Chao 1972 carried out a detailed cytological investigation on Papillum orbiculaire and demonstrated that the digital part of the integuments, by dissolution of its cells in situ, secrete a mucilaginous substance into the micropyle, which provides a way of least resistance for the pollen tube and guides it toward its ultimate destination. This is water soluble carbohydrate and helps in pollen tube growth both mechanically and chemotropically. In this slide, we see the all three types of fertilization based on the entry of pollen tube into ovule: porogamy, chalezogamy, and mesogamy. A special structure which facilitates the entry of pollen tube into the ovule is called obturator. It forms a sort of bridge for the pollen tube to reach the ovule. After fertilization, the obturator shrinks and disappears. In family Loranthaceae, there is no structure like an ovule. The embryo sacs undergo remarkable elongation and meet the pollen tubes at some point in the stylar canal or stigma. एंट्री ऑफ देन आता है नेक्स्ट टॉपिक, एंट्री ऑफ पोलन ट्यूब इनटू द एम्ब्रियो सेक। इररेस्पेक्टिव ऑफ द प्लेस ऑफ एंट्री ऑफ पोलन ट्यूब इनटू द ओव्यूल, इट इनवेरिएबली एंटर्स द एम्ब्रियो सेक एट द माइक्रोपाइलर एंड. The tube enters at the apex of the filiform apparatus and after growing through it arrives in the cytoplasm of the synergid. Which one of the two synergids a pollen tube would enter seems to be predetermined. Mostly the penetrated synergid starts degenerating before the arrival of the pollen tube, but after pollination. This receptive synergid is readily recognized after it degenerates because it lacks plasma membrane and tonoplast. Increase in GA3 concentration in the ovules as a consequence of pollen tube pistil interaction has been suggested as possible cause of the breakdown of synergid in cotton. Next, the contents of pollen tube are discharged in the synergid and the tube does not grow beyond it in embryo sac. In plumbago, the pollen tube enters the synergid-less female gametophyte through the filiform apparatus at the tip of the egg. It penetrates the egg wall and discharges the sperms and the tube nucleus between the egg and central cell. The pollen tube discharge. It includes two sperms, the vegetative nucleus, and a fair amount of cytoplasm. The cytoplasm released by the pollen tube and that of the synergid hardly show any mixing. They remain as two separate entities. The synergid cytoplasm is confined to the micropylar end, and that of pollen tube is restricted to the cellulose end of the cell. The cytoplasm released from the pollen tube undergoes immediate degeneration, then sperms. The two sperms in a pollen tube often change their shapes. They are cells, each bound by plasma membrane. In tube, the two sperms are usually placed close to each other. Cytoplasm contains the usual cell organelles. Nucleus contains a distinct nucleolus. In this slide, a portion of penetrated synergid shown by Jensen and Fisher 1968. Post pollination changes in the embryo sac. The synergid, which is visited by pollen tube, degenerates faster than the other. The former is called degenerating synergid and the latter is known as persistent synergid. There are two types of synergids. Some characteristic features of degenerated synergids are: the large chelazal vacuole disappears, the nucleus and the nucleolus are somewhat flattened, and the nuclear membrane disappears. The organelles get disorganized, crystals appear in the cytoplasm. Next, X bodies. Two darkly staining oval bodies in the synergid which has received the pollen tube discharge. Since the nature of these bodies was not clear, they were denoted as X bodies. From time to time, these X bodies have been variously interpreted as remains of the vegetative nucleus, remains of the synergid nucleus, cytoplasm of the sperms, adjacent nuclear cells, remains of the degenerated megaspore, etc. Based on their shape and distribution in the synergid and the fact that they contain DNA, one of them as remains of the synergid nucleus and the other as remains of vegetative nucleus. Next, double fertilization. S. Navastin first to show that both the sperms released by a pollen tube are involved in fertilization. So, in this case of angiosperm fertilization involved two sperms. They fertilize two different components of embryo sac. The phenomena is unique to angiosperm and is called double fertilization. The nucleus of one sperm fuses with the egg nucleus syngamy, and that of other migrates to the central cell where it fuses with the polars or their fusion product, the secondary nucleus. Most of the plants have two polar nuclei. Consequently, the second fertilization involves fusion of three nuclei, and the phenomena is called triple fusion, which is characteristic of angiosperm. Passage of the sperms. The sperms are released in the synergid as intact cells, but according to general belief, only their cells nuclei migrate out of it. However, evidence has been presented which suggests that the male cytoplasm is also sometimes involved in fertilization. Russell 1980 demonstrated the transmission of cytoplasmic organelles of the male gametes during gametic fusion in plumbago. Similarly, Case 1981 has shown the entry of male cytoplasm into the egg in barley. Next, the mode of sperm transfer is a matter of speculation at present. According to Jensen 1973, one of the sperms come in contact with plasma membrane of the egg cell, while the other contacts with plasma membrane of the central cell. The membranes at the point of contact dissolve and the sperm nuclei are released. One in egg and other is in central cell. After entering their destinated cells, the male nuclei are passively carried out along the cytoplasmic stream to the egg nucleus or to the polar as the case may be. The entry of one of the sperms into central cell is preceded by the entry of other sperm into egg. So, first sperm entry into central cell and then entry into eggs. Thus, the first sperm nucleus reaches the egg nucleus before the second sperm nucleus reaches the polars. However, the fusion of egg nucleus and sperm nucleus takes much longer than the fusion of the male nucleus and the polars. In this diagram, summary of sperm transfer in embryo sac, we so see all these phenomena. Then syngamy. Fusion of egg nucleus with sperm nucleus is called syngamy. Gerasimova Navaschina 1960 has described three types of syngamy. According to her, at the time when two nuclei come in contact with each other, the egg nucleus is in state of deep mitotic rest, whereas the male nucleus is at a telophase of the previous mitosis. These three types are pre-mitotic, post-mitotic, and intermediate. Unusual features related to fertilization as are follows. Polyspermy, a situation where more than two sperms are released in the embryo sac due to formation of more than two sperms in a pollen tube or due to penetration of an embryo sac by more than one pollen tube. Polyspermy may bring about fertilization of egg by more than one male nucleus, or the supernumerary sperms may fertilize other components of the embryo sac such as synergids or antipodals, etc. In the embryo sac, which received two or more pollen tubes, the sperm nucleus fusing with egg nucleus may be derived from one pollen tube, and one fusing with the pollers may be derived from another, called heterofertilization. Next, persistent and branched pollen tubes. In angiosperms, the pollen tube is normally an unbranched structure which collapses soon after fertilization. Some reports, however, claimed that rarely the pollen tube may persistent for as long as 3 weeks. In cucurbita and some members of the Onagraceae, branching of the terminal portion of the plant pollen tube has been observed. According to Wilms, branching of pollen tube is probably a post-fertilization phenomena. So, in this video, we have discussed on remaining aspects of fertilization like pollen tube entry and double fertilization, triple fusion, post-fertilization changes, and other unusual features related to fertilization. Thanks to all